Ladies and gentlemen, I'm going to talk about complicated food webs in tropical forests and the way we can study them by experimenting with them. There are ecological mysteries concerning the forest, especially why the world is green and mostly forested, and why tropical forests have so many species of trees. We can rephrase these questions by wondering what stops herbivores from eating the world's vegetation into the shape and form of a golf course, and also what stops trees from outcompeting other trees, making the world's vegetation look like a tree plantation. Well, the answer is in control of herbivores, which can be either by natural enemies, top down, which would mean the world is green but dangerous, or by their own plants, by resources, bottom-up control, which means the world is green, but the green stays awful. For plants, the options are more limited. It's unlikely that the diversity of resources will lead to such tree diversity, and so we suspect the top-down control by natural enemies. When we are talking about the diversity of trees, then the obvious question is how many tropical trees are there? And that question was actually not so obvious to many botanists. It's only in the 2015 paper by Ferry-Slick, which answered the question, and even that with quite a range between 40 and 53,000 tree species. It was actually entomologists, especially Terry Irwin, who were first asking how many trees are there, because Terry Irwin wanted to estimate the number of tropical insect species, and so he had a good idea about the number associated with individual tree species and wanted to multiply it by the number of tropical tree species, and found that nobody actually knows how many are there. So he was a curator at the Museum of Natural History at the Smithsonian, and so he asked his colleague Richard Ide next door, and the botany curator, and he also didn't know. So Ide called a colleague, Richard Howard, on the phone, and after some minutes of intense thinking, Richard Howard ventured a guess that there might be around 50,000 tree species. Terry Irwin used that, and although his study, which led to a quite shocking estimate of 30 million insect species, has been questioned and examined and challenged by many colleagues, including our own work. While we as entomologists were examining his claims about the host specialization and other insect aspects, nobody really questioned the 50,000 species of tropical tree, and everybody forgot that it's based on a friend on a phone. Until Ferry-Slick and his colleagues decided to resolve the matter. And we might wonder how many botanists you will need to change a light bulb, but we certainly know that you need quite a few botanists to count the tropical trees, as you can see from the authorship of the paper. And quite disappointingly, they actually came to the conclusion that the original estimate was more or less right, and we have more or less 50,000 tropical tree species. However, that is not the main mystery. The main mystery is why are there so many tree species at a very small geographic ranges. For instance, this is our 50-hectare forest dynamics plot in Papua New Guinea, which I will still be mentioning. And there, in a typical 20 × 20 meter plot, there are 85 species of woody plants. And so that mix with species having heterospecific neighbors, that is something we need to explain as ecologists. And that explanation was suggested by Daniel Janzen and Joseph Connell, and is known as Janzen-Connell hypothesis, published in 1970 and 71 independently. Interesting example how some ideas simply float around, and two people independently took them. Then Janzen mostly used insect-plant interactions model, while Joseph Connell the marine systems, but explained on the Janzen's picture. This is the dispersal kernel of seeds from a parent tree, which is pretty much always this shape. Lots of seeds falling close to the tree, and then their numbers declining rapidly. But because of the contamination of seeds and later seedlings and saplings by either herbivores from parent tree or pathogens, especially pathogenic fungi, then the probability of seed survival is increasing with the increasing distance and decreasing density of seeds from the parent tree. And so combining these two functions, we will get a recruitment not close to the parent tree, but at some distance from it, exactly the pattern we see in natural forests. So for plants, it's vital to get away from their parents, which might be also a universal law of life. So with this hypothesis, how can we test it? How can we simply prove it conclusively and be done with it? Well, we can look at spatial variation in natural vegetation, taking into consideration the age of the plants. We can substitute space for time, or we can actually be patient and look how forests change over time. And that can be on natural vegetation, but we can also plant our own experiments with different plant densities and look at how it works. And finally, we can also manipulate the natural enemies of plants on the native vegetation, or again on the planted systems. So when we start with the observation approaches, which actually dominated the test of Janzen-Connell for the first incredible 40 years, interestingly, neither Jensen or Connell showed any intention to empirically test their proposition. However, Steve Hubble and Robin Foster did try that by starting the tradition of 50-hectare forest dynamics plots, monitoring all plants with the stem diameter at least 1 centimeter. So that was in 1980s, and at that time, their first plot was at Barro Colorado Island in Panama, and it was a quite unusual, quite surprising development, because at the time the gold standard of tropical botany was studying 1-hectare plots with DBH 5 centimeters, and that will give you about 1,000 stems. They proposed 300 times bigger sample size, and we know that if you propose to your colleagues that they should sample 300 times more, then they will laugh at you and then they will reject your grant. And so it was always mysterious how they managed to get the National Science Foundation grant, which funded the plot. That was actually clarified in a book by Jan Sepp, where he notes that the botanists were indeed skeptical on the National Science Foundation panel, but there was an ornithologist who pushed the proposal through, and because of that, we have what is probably the most successful and productive ecological collaboration of the current time, and that is the Forest Geo Network, which grew to 67 plots, 6 million trees, and 13,000 species. Includes also our plot Wanang in New Guinea. I will only briefly mention one study, and that is the analysis of spatial distribution of plants, which is decomposing the distribution of saplings into the effect of conspecific negative density dependency of adults and heterospecific negative density dependency of adults and saplings, and using all these 50-hectare plots across the globe, and comes to the conclusion that the strength of conspecific negative density dependency is increasing towards the tropics. When we look at the individual communities, then the species which are locally rare are especially strongly limited by conspecific negative density dependency. So that would explain the abundance patterns, but interestingly, that pattern can be seen only in the tropical communities, and it disappears in the temperate zone. However, when we really want to see what's happening, then the philosophy of the food web research is to somehow disturb the complex food web, kick them around and see what happens, because otherwise, with such a complex systems, circumstantial observational evidence will never be conclusive enough. So we are getting to enemy manipulation in natural vegetation, and that has been done by Robert Bakshi and Owen Lewis at Oxford a few years ago, where they were using insecticides and fungicides to spray seedling communities in the forest, and found that one type of fungicide was indeed effective. With fungicide, the diversity declined, so that means that fungi should have diversifying effect on vegetation. And when looking at experiments with different initial seedling density, then in natural conditions you ended up with constant seedling density, so that means the higher initial density, the higher mortality, while the fungicide released that negative density dependency and led to a positive correlation of initial and final seedling density. So this was evidence for fungicides and therefore for fungi in the forests. In our study, we have looked at slightly different problem. We have looked at, again, effect of fungicides, insecticides, also exclusion of predators, ants by poisoning them, and then bats and birds by these nets, and also effect of additional generalist insect herbivores added to the system. And all this was done in these plots in New Guinean rainforests, and looking at the initial stages of secondary forest succession. So that's a bit of a change of the focus, because based on the ecological theory, there should be no Janzen-Connell effect in these initial stages. We all know from the textbooks that there are extremely specialized pioneers which are maximizing the growth rate because they have to compete with other pioneer species, and they don't really care about being eaten by herbivores. So we tested this proposition, and this is how it looks in the lowland rainforest, spontaneous secondary succession after 1 year with a healthy, diverse pioneer community of plants. And this was done with my PhD student Piotr Schäfer, and we found actually that it's insects which are diversifying the initial stages of vegetation, so that insecticide application led to lower diversity. And they also change the species composition of vegetation, unlike fungicides or exclusion of predators, which did not. And finally, they also improve the predictability of the course of succession. We had actually a big problem that succession tends to be influenced by random factors, what is in the seed banks, what is in the immediate neighborhood. And so our 6 replicates of these blocks proved to be not really a very strong test, and probably some of the lack of significance is due to sample size, but the insecticide actually, the effect of insects actually increase predictability of successional trajectories. So if insects control vegetation, what controls insect herbivores? The exclusion of vertebrate predators and ants again led to increased diversity of, in this case, herbivores. So there is again diversifying effect of predators on herbivores, but to our surprise, there was no effect on herbivore biomass or abundance. And also there was no effect whatsoever on non-target predators, which were mostly insects other than ants and spiders. And so we have to conclude that there is not very strong top-down control on herbivores. In fact, the effect of our treatments, our exclusion of predators on plants and herbivores was positively correlated, which was opposite to our expectations. If herbivores are enemies of plants, then what is good for herbivores should be bad for plants, and vice versa. The same goes for herbivores versus non-target predators. But in fact, it looks like the resources, the bottom-up effects are dominant in the system as far as predators and their effect on herbivores are concerned. A few comments on this experimental system. This is actually currently second stage of the experiments where we'll be manipulating specifically the development of vegetation, and again using insecticides to look at the effect of insects on that. And this kind of experiment is possible in Papua New Guinea because the indigenous communities are living in the rainforest and they are practicing swidden/burn agriculture. And so it's easy to convince them that instead of planting sweet potatoes, they should help us to do experiments, which is actually also bringing better income and not contributing to deforestation, because we are using these garden systems. In this case, we are trying to reduce this variability in succession trajectories by scraping the topsoil, mixing it for all the experimental plots, and then spreading again. And before we do that, we remove the secondary vegetation and also sample it for insect communities. And here we have the plots ready again. So this is an example of such a secondary succession on former food garden in the center of the picture, and this is how we make it into experimental plot. And the whole system allows us this kind of almost perfect design of secondary plots within the primary forest matrix, perfectly mimicking the threefold gaps for the experiments. So that's an advantage of indigenous communities and work with them. Probably hard to do in some other countries, such as neighboring Australia. And then we go to the next option, and that is the enemy manipulation in planted vegetation. There, again, in Vanang 50-hectare plot, our colleague Yves Basset assembled diverse seed plots with 23 species. Each seed was marked by a cocktail pig. We were the biggest buyers of cocktail pigs in the whole province, with some consequences to our reputation. And these are the results. It looks like in at least some trees, the insecticide again have a significant effect, although the final data are still being analyzed. This is another huge manipulative experiment, which was sort of unintentional, and this time it applies to fungicides. This is Heva brasiliensis, the rubber tree, which is, as the name suggests, native to the Amazon. And in the 1928, the Henry Ford didn't like that his automobile business was dependent on British rubber from the Southeast Asian plantations, and so he founded Fordlandia, a town in the middle of the Amazon forest, and planted huge areas of hevea, which were immediately destroyed by fungus. So this was basically Janzen-Connell hypothesis tested 50 years before it was published. And the experiment failed not only biologically, but also socially. The local records say that the native workers grew tired of American food and revolted in the town's cafeteria, chased away the managers and townsfolk into the jungle. Apparently, you can still visit ruins of Fordlandia in the Amazon. Now, all these experiments are good evidence, but they are kind of black box experiments. We are removing fungi, don't know which ones. We are removing insects, we don't know which ones. So as biologists, we really need to find out specific interactions there. This is example of such study rearing seed predators, again in Vanang, a plot from more than 300 kilograms of seeds, and we found very complicated food webs with the plants and their herbivores, and then they have their own parasitoids. But when we look at the quantitative aspects, this is the frequency of Curculionid weevils per fruit. We can see that with 326 plants tested, a large part of them have no species at all, and even those who do have some, then the rate of attack is too low for any Janzen-Connell effects. So at least for this, minority of trees is subject to that mechanism. So we need to basically search further and come with specific mechanisms. We can also turn Janzen-Connell around and ask what happens if a seed benefits from the proximity of the parent tree. That can very well happen, for instance, for mutualist interactions, and we have a very good candidate, which is ant plants, where the protective effect of ants on survival of seedlings can spill from the parent tree, and in that case, the seed distribution will the clustered effect of seed distribution will be enhanced by improved survival in proximity of the tree, and so we will get extremely clustered vegetation pattern. That actually can be illustrated in so-called devil's gardens in the Amazon, where in the middle of nowhere suddenly there is a monospecific stand of ant plant Duroia hirsuta, and recently Megan Frederickson at Harvard demonstrated that the ants are actively destroying any seedlings and saplings of plants other than Duroia hirsuta. So this is exactly the effect of the reverse Janzen-Connell, and some of these devil's gardens are more than 800 years old. Fortunately, when they are getting bigger, then ants cease to be able to manage it, because otherwise we can easily imagine that in the extreme it would be possible for Myrmelachista, Schumani, and Duroia hirsuta to convert entire Amazon lowland into a monospecific growth of Duroia hirsuta. So we have seen that herbivores can have diversification effect on plant communities on local scale. At the same time, we know that the tropical maximum in diversity applies to both plants and herbivores. So that leads to a question whether there is some interaction on that level, whether, for instance, the herbivores are diverse, especially in the tropics, on their own, or whether they are just following increased offer of plant species as the host plants. So for that, we have done surveys of 6 food webs between plants and herbivores, 3 temperate zone, 3 tropical, at different continents. We used canopy cranes or elevated platforms, or cutting down the forest, which was earmarked for logging anyway, not contributing to deforestation. And in Papua New Guinea, we even built our own canopy crane, which was a rather adventurous experiment, which I don't really recommend to everybody. And then with a tropical sample, we were able to subsample it to mimic the temperate zone much species poorer sample. So we selected trees of the same number, size, and number of species as in the temperate zone, and then compare the structure of the herbivorous community. And we can do that and then we can model these effects also for vegetation plots globally. So when we look at the vegetation plots, there is indeed a latitudinal trend in trees, and then when we use our data to predict what we expect from miners purely based on the changing plant diversity, then we got again a strong latitudinal trend, and our empirical data from temperate zone communities pretty much fit the trend. So we can preliminarily conclude that miners are simply following plant diversity. There is nothing special about the tropics in terms of their diversity. Ants are different. They don't follow plants as closely, and the temperate zone ants are seriously under the predicted diversity effect. So there is actually a genuine tropical temperate zone gradient in ants independent of plants. Okay, so that is all from me, and I would like to thank especially my New Guinea collaborators who contributed to many of these studies from the New Guinea Binatang Research Center.